So, hello everyone. This is the very second episode, if you will, of our Western Biology Evolution Seminar Series. I am your host, Matheus Sanita, and I'm thrilled to have here with me today, Joanna. So, Joanna Rocha is a Portuguese speaker fellow of mine, and I'm very happy to have someone who can speak Portuguese with me today, though, of course, the talk and the Q&A will be all in English. So let me tell you a little bit about Joanna. Joanna is an evolutionary biologist and currently a postdoctoral researcher in the Department of Integrative Biology in UC Berkeley. She received her PhD in biodiversity, genetics, and evolution, having been advised by Raquel Godinho at the University of Porto and Rasmus Nielsen at UC Berkeley. When she's not doing research, she enjoys being in close contact with nature and sharing all the amazing wonders our planet has to offer with the people that she loves. She was born in Portugal, and she speaks fluently Portuguese, English, Spanish, and a little bit of French. And she tells her friends and audience they can feel free to contact her in any of those languages. And today we'll be learning from her a little bit about the genomic signatures of the life in the deserts. So, Joanna, please feel free to take it away.
Thank you so much, Matheus, for such a nice and kind introduction. About the languages part, I think that's no longer needed. I guess these days with ChatGPT, everyone can just literally translate very easily emails and have it right for them. So, but anyway, yeah, if any excuse to practice a different language, please feel free to approach me, and I would love that. So I will be now present my sort of an overview of my PhD dissertation work, and that is entitled Life in Deserts and the Genomic History of North African Foxes. All right, so there are two questions ever since I started grad school that I really wanted to address. One, how species adapt to new environments. And secondly, as species diverge and environment change, how adaptive genetic variation is maintained, or for how long can beneficial alleles survive? And extreme terrestrial habitats are remarkable settings to address these questions because they are home to populations or species that likely have been the target of natural selection. So the last decade of genomic research on mammalian species inhabiting the cold high Arctic and the hypoxic high altitudes, for example, consistently have been able to identify or pinpoint genes associated with adaptation to life in these extreme conditions. But collectively, they also led to very exciting discoveries on the origins of adaptive genetic variation. So, for example, sometimes we have same or different solutions to different selective pressures. Mammalian research has well established that integration is very pervasive in nature, and it can be a really important source for adaptive genetic variation. And this sometimes involves long diverged species or even unidentified lineages. So it's not just between pairs of closely related species. But ancient polymorphisms maintained by, for example, long-term balancing selection can also explain shared adaptive genetic variation across speciation events. And most examples of long-term balancing selection known in the literature, for example, come from genetic variation in the form of SNPs, single nucleotide polymorphisms. There are, in fact, many different types of genetic variation. And this example that I'm showing here on the right, this is a case where structural changes in genes, in this case deletions, that date back before modern humans split from their ancestors. And interestingly, Akil et al. Found that these deletions, these ancient genes, ancient deletions persisted in modern humans to date, not because of integration, but because they have been maintained across speciation events by balancing selection. Now, back to extreme terrestrial environments and why they are so exciting settings to study and address natural selection. Deserts make up a third of our planet's land surface and are some of the most climatically challenging and driest environments on Earth. But at the time I started my PhD, the genetic basis of desert adaptation was still an open question, actually. And when I talk about deserts, I should make it clear that there is no such thing as a single desert. Deserts are quite different. But I guess that if I could pinpoint what is the main challenge or something that is common across all deserts, is that the main challenges for species or populations are maintaining body temperature and preserving water. And this seems like an intractable problem for species that, for example, depend on water loss mechanisms for thermoregulation. But the truth is that deserts harbor spectacular life forms that thrive under extreme conditions. And in fact, part of my dissertation work established this hypothesis of aridity as one of the main selective pressures driving repeated evolution of similar phenotypes in different desert mammalian species. And this is a raw illustration of the relationship between aridity and maximum urine osmolality, which is a form of urine concentrating ability. And this basically shows that mammals with small body mass can concentrate more urine than increasingly large mammals. And this is a very well-established notion that urine concentrating ability is negatively correlated with body mass. But most importantly, if you look into these broadly defined body mass categories of evader, evaporators, and endurers, you will see that species inhabiting arid environments can actually maximize urine concentration well above the levels of their non-desert counterparts. And so as genomic studies continue to search for the genetic basis of desert adaptation, maximum urine concentrating ability, for example, might be one of the primary physiological traits that lends itself to more detailed genetic analysis. However, one thing that I've noticed as I was doing through my PhD is that most desert studies are limited by the expense of research in remote arid regions, and most importantly, the logistical issues that challenge the possibility of gathering integrative data sets or even more complex experimental designs. We also know that there's a lot of limitations that are associated with lack of detailed knowledge on the causal mutations underlying desert adaptive traits. And this may be in part due to sampling size issues, but also in part that genetic variation that could be associated with phenotypes may not be always found segregating in nature. And so there is a lot of challenges of addressing desert adaptation and studying how life can thrive in deserts from a holistic, integrative, multidisciplinary, evolutionary biologist perspective. But this is a review that we did for Trends in Ecology and Evolution, where we encourage research on systems where methods for genetic mapping or functional studies can be integrated with selection studies. But we also recognize that model organisms like mice or doing research on humans shouldn't be the only focus when we think of desert adaptation, because these systems actually represent a narrow set of organisms, and there is a need for more studies at a wider taxonomic range. And there are many desert species that are not the standard ones that are like desert mice, camels, or humans that are waiting to be explored and understood. And so the goal of my thesis was not just to advance our general understanding of adaptive evolution in deserts, but actually contribute with understanding of how desert adaptation operates from the perspective of wild populations and non-model organisms. And so I guess in the first part of my PhD, I try to address this at a larger scale with a revision work that aims to understand the genetic basis of mammalian desert adaptation, and a meta-analysis that is focused on the role of climatic factors driving the evolution of desert adaptive traits. And if you're curious, I encourage you to go and search and check out these works, because mostly they provide the basis and motivation for the most integral part of my grad school life and what I'm here to talk about, which is to zoom in the Sahara Desert, which is the largest hot desert on Earth, and apply a genomic evolutionary genomics approach to investigate the genetic basis and the evolutionary processes underlying desert adaptation in a model group of North African foxes. Now, why foxes? Well, foxes of the genus Vulpes are the only group of canids with geographic distributions that fully overlap some of the most extreme deserts on Earth. In fact, just by briefly looking into the Vulpes, which is a genus Vulpes phylogeny, you will find pairs of closely related species that diverged into different environments, either arid versus Arctic, lowland highland, mesic Arctic. And this provide an unprecedented opportunity to study adaptation to divergent or different selective regimes. But you will also find cases of repeated adaptation to similar environments by long diverged or more distant lineages. And this offers a very unique opportunity to study mechanisms underlying shared adaptations and at distinct spatial temporal scales or evolutionary scales. Now, North African foxes in particular constitute an entirely new system to address the historical events and evolutionary processes at play during colonization of arid environments and adaptation to new climatic conditions. For example, the Rupel's fox, represented here, is a desert specialist species that locally overlaps with a distant related congenitor, the fennec fox, known for its characteristic oversized or super large ears compared to the head size and body size. And the two species share a huge part of the geographic distribution in North Africa. And both species are actually remarkably adapted at coping with extreme temperatures and water scarcity at the Sahara. But the Rupel's fox is actually more closely related to the red fox, which is a generalist species that is found in a wide variety of habitats across the northern hemisphere. Here representing in green for the red fox and the reddish, pinkish color for the Rupel's fox. The two species are actually presumed to be of Eurasian origin, sharing common ancestors with Asian endemic fox species and the Eurasian North American Arctic foxglove before divergence from the fennec fox, and also the pale fox, a basal species endemic to the Sahel. Now, though red fox and Rupel's fox diverge into contrasting environments and actually differ fundamentally in ecology, behavior, and physiology, the two species locally overlap at the northern fringes of the Sahara and Middle Eastern deserts, where red fox desert morphs have actually been described. And notably, a previous study alluded to past introgressive hybridization events between Rupels and red fox in North Africa. So, okay. If this was the case, the integration between Rupels and red fox, this would be in line with previous studies that have actually shown that integration is pervasive in canids. And mostly that integration could be a really important source for adaptive genetic variation between either sister species or sometimes even involving unidentified canid lineages. And so the Sahara and its northern outer edges represent a natural laboratory for addressing questions regarding red fox dispersal into North Africa, ancestral hybridization events, and natural selection processes at different spatial temporal scales. Now, while interspecific analysis, so analysis within species, may offer a detailed perspective on recent or ongoing selection and on the role of past climatic oscillations in driving desert adaptation, in what we could consider to be short evolutionary timescales, comparative analysis among desert-dwelling species have the potential to elucidate the evolutionary routes underlying the origin of adaptive genetic variation, which are all still outstanding questions in the desert. So here represents what would be addressing interspecific analysis, where we would be comparing, for example, red foxes from Eurasian backgrounds, from non-desert regions, and red foxes, let's say, from North Africa, which are currently inhabiting the outer edges of the desert, and a comparative analysis where we could, for example, focus on the red fox from North Africa, the sister species of the red fox, which is the Rupel's fox that inhabits the North African Sahara, and the more long diverged fennec fox that inhabits also the North African Sahara. And so to explore all these possibilities, we took advantage of a set of tissue samples from roadkill animals for several fox species and complemented these with field expeditions and sampling in captivity. And these efforts not only allowed us to maximize sampling size, but also allowed us to collect fresh blood samples for physiology and expression analysis. And then we were able to use this data as a potential complement to genomic analysis. And so our sampling efforts actually allowed the sequence of complete genomes of 82 foxes to approximately 4.5x coverage. And this comprised Eurasian and North African populations of red fox and a total of 4 species. We then used a set of statistical tools for low to medium coverage datasets and started by briefly addressing species relationships and population structure, with a particular focus on sister species, Rupel's fox, and the red fox from Eurasia and North Africa. And so first, we found, using phylogenetic trees and admixture analysis, that the sister species, Rupel's fox and red fox, form 2 distinct lineages or clades, outgrouped by the Arctic fox and only then by the fennec and the pale fox. We also found the red fox populations are first separated into Eurasia and North Africa, and then Eurasian populations separate into Europe and Asia. And this suggests that gene flow has been more limited between North Africa and Eurasian red foxes than among Eurasian populations, which would be consistent with the scenario of red fox isolation in North Africa. Now, the fact that there is no evidence for recent or ongoing admixture between North African foxes does not actually exclude the possibility of ancient hybridization after divergence. And so to further investigate integration and understand when Rupel's and red fox diverged, we modeled a joint demographic history of the 2 sister species in North Africa using GADMA and DAVI, and our base model for migration after the divergence approximately 576,000 years ago. Of course, I should say here that there is an inherent uncertainty on the genome-wide mutation rate estimate and the variance in generation time, because we are assuming here the mutation rate to be that the same of the dog, and the generation time to be that of 2 years. But still, even with that uncertainty, we do find that the divergence time estimates between the 2 species is in accordance with the earliest fossil records of red fox, and within the coalescent time interval of 400 to 700 thousand years ago, that we estimated using mitochondrial genomes from several vulpa species. We then used an Abba-Baba test for these statistics to look for integration among species, and we did find an excess of derived alleles shared between North African red foxes and Rupel's relative to Eurasian red foxes, which is a signature of integration and is also consistent or supported with our demographic modeling. But perhaps what was truly unexpected here is that when we tested for integration between core desert-dwelling species, we found that there was significant evidence for excess derived allele sharing between Rupel's fox and more divergent fennec fox. And this was truly unexpected. Now, our hypothesis is that maybe integration among locally overlapping and ecologically similar species could have facilitated evolutionary responses to life in arid and hyper-arid regions at different evolutionary scales. And so, motivated by this hypothesis, we further explored each of these integration events by calculating F statistics on sliding windows to identify specific regions or fractions of the genome that were potentially shared by integration. And so, using the fraction of admixture statistic, or F of D, we found several genomic regions with overlapping genes, presumably shared between Ruppel's fox and North African red foxes. We then compare the average genome-wide tree with a tree topology for some of our outlier genes in the F statistics and focus on cases in which gene species discordances had this specific pattern where some North African red fox individuals appear to be more closely related to Ruppel's fox than Eurasian red foxes, which would be consistent with a scenario of integration into North Africa red fox from Ruppel's. And this was the case of SLC6A16, a poorly studied sodium transporter. This gene had the second strongest signal of integration in FD, and consistent with an integration scenario, the divergence across this region was below the 0.5% of the genome-wide empirical distribution between North African red fox and Ruppel's fox. Though linked selection in ancestral population can lower the value of the XY, and we did not explicitly test which this hypothesis is more likely. Genome-wide scans for introgress regions between Fennec and Ruppel's fox identified this unusually large region, 25 megabase region, that is shared between these two distantly related species. Now, this whole region actually mapped to the dog's chromosome 13B, and past chromosome correspondent analysis actually matched this arm to different chromosomal arms in the red fox, the Arctic fox, and the Fennec. And so our explanation for this huge region and for this remarkable outlier region of loci that are tightly linked in strong linkage disequilibrium is that this is probably the result of a structural rearrangement, like a translocation of a chromosomal arm or an inversion. Now, consistent with an integration scenario, tree topologies for this 25 megabase region and unambiguously grouped all Ruppel's fox as sister species to the Fennec, which suggests that these structural variants are fixed in Ruppel's fox. However, the pairwise divergence between the two desert-dwelling species at the region is not much different from the genome-wide average. And so our hypothesis is that integration did not happen directly between the Fennec and Ruppel's fox, but instead from an unknown Fennec-related species. We also note that the data does not allow us to distinguish between integration and the hypothesis of, for instance, an ancient transspecies polymorphism, with a Fennec-related divergent recombination suppressed haplotype seemingly becoming fixed in Ruppel's fox and then repeatedly lost, for example, in the Arctic fox clade and the red foxes. Now, the interesting thing is that when we did enrichment analysis in the 25 megabase outlier region, we identified several genes or gene ontology terms that were actually overrepresented. And this included genes coding for sulfotransferases, involved in stress response, metabolic fasting, thyroid hormone metabolism, and neuromodin receptor that is involved in temperature homeostasis, regulation of heat generation, and feeding behavior. All of these that have like potential phenotypic relevance for desert life. And this suggests that shared genetic variation with other resident desert species, either in the form of integration or shared ancestry polymorphisms, as well as between more recently evolved species, might be a potential source of genetic variation on which natural selection can act upon. But we also wanted to understand another perspective. We wanted to understand the perspective of not just integration between species or shared genetic variation between species, but what makes these species uniquely adapt to life in the Sahara. So we wanted to explore unique adaptive histories of these different fox species. And so to do this, we approach this at a population or interspecific level by modeling the joint demographic history of North African and Eurasian red foxes. And we wanted mostly to address dispersal events into North Africa by the red fox. Now, what we found is that the model with the highest composite likelihood dates the divergence of the two species to approximately 78,000 years ago. Now, this is probably in a period that has been associated with the Green Sahara, and it could have enabled an ancestral red fox population to colonize North Africa and expand to regions that were previously uninhabitable for the species, setting the stage for divergence from Eurasia and integration with North African Rupel's fox in subsequent arid regions. Now, North African red foxes that were living in increasingly arid environments at the Saharan northern edge for numerous generations, likely received variants from rupels that have made facilitated survival in these habitats, as I've shown before in the history of integration between rupels and North African red foxes. But red foxes may also have faced unique selective pressures to increasingly arid regions of the Sahara over the last couple of thousands of years. And so to investigate signatures of positive selection specific to North African red foxes, here's what we did. We looked at two lines of evidence of selection: genomic regions that were either highly differentiated and regions with reduced divergence in the focal population, which is North Africa, relative to Eurasian red foxes and Rupel's fox. This is also known as the PBS statistic, or the minus log 10 of alpha pi. We then focus on the most extreme values of the empirical distribution of these two selection scans and identified our overlapping genes. And then the most interesting thing is that our top hits overlapped with candidate genes associated with actually interesting and potentially relevant traits for life in arid regions. This is the case for SLC12A2. So knockout mice for this gene, which encodes for a sodium chloride transporter, have actually reduced saliva and sweat production. And we did find a strong selection signal in SLC12A2 as to be potentially related with selection for changes in expression, because this gene is actually less expressed in North African foxes and Rupel's fox, for example, than red foxes. And so our hypothesis is that selection potentially acted on this gene in North African red foxes to reduce the amount of evaporative water loss through mechanisms like panting or sweating, which are the main mechanisms for cooling in canids. Another highly differentiated and low diversity gene is TRPA1, which encodes for a thermal TRP channel protein that is gated by noxious cold or heat. Now, several thermal TRP sensors have been reported to be under selection in Arctic sled dogs and mammoths and implicated in increased cold tolerance. Notably, the gene coding for thyroid adenoma-associated protein, THADA, which is involved in heat production and widely associated with climate adaptation in humans, is also under selection in North African red foxes. And so, though arid regions in North Africa are classified as quote-unquote hot, they may experience extreme daily temperature oscillations with drop-off temperatures at night. And so North African foxes are actually nocturnal. And we hypothesize, in the light of these results, that reduced thermosensitivity to both extreme cold and heat could be adaptive in arid regions with high isothermality. So collectively, these results suggest that North African red foxes have unique adaptations related to water retention and tolerance of extreme temperatures, and that they may have increased survival chances in the increasingly arid regions of the Saharan fringes over the past 78,000 years. Now, this raises the question of what signatures of selection are to be found in other fox species adapted to life at the core of deserts for successively longer periods of time, such as the case of the Ruppel's fox, and of course, our friendly, charismatic fennec fox that most people are probably well acquainted with. So to address this, we performed a genome-wide selection scan implemented in a software called Ohana that can be applied to several ancestral components. And what we found is that there are signals of selection in several regions harboring relevant genes related to temperature homeostasis and thyroid hormone metabolism, thermotaxis, and response to UV radiation in Ruppels. And what I would like to call your attention here is for this gene called WFS1, marked in bold, that is related to renal water homeostasis. Now, knockout mice for WFS1 show a very strong impairment on vasopressin secretion. And vasopressin is the main hormone for stimulating water reabsorption at the kidney. So these deficient WFS1 mice actually have decreased ability to concentrate urine under water deprivation. And so our hypothesis here is that mutations changing expression of WFS1 may explain increased urine concentrating abilities in Ruppel's fox. Now, consistent with this hypothesis, we noted that this gene is significantly more expressed in Ruppel's fox than red foxes. And by looking into physiological parameters on blood samples, we also found that levels of capeptin, which is a biomarker for vasopressin, were significantly higher in Ruppels compared to red foxes, further pointing to what we would expect to be an increased water retention ability in Ruppels relative to both North African and European foxes. Now, another thing that we note is that WFS1 gene is within this really high 25 megabase region that seems to be shared between Ruppel's fox and a fennec-like donor. So this suggests that Ruppels inherited increased capacity to retain water under dehydration from a related and as yet to define desert species that is very closely related to the fennec. Fennec also have higher capeptin or vasopressin levels and are known to concentrate more urine than most non-desert canids, and in this case, than red foxes, for example. And the gene with the highest selection signal in fennec is actually the KIREL1, which encodes for a protein produced in kidney podocytes that plays a significant role in the normal function of the glomerular permeability. And so KIREL1 may be one of many candidate genes of desert adaptation identified in this study. And all of these genes that have been candidates of natural selection and potentially underpinning adaptation in foxes, they all remain promising targets for future studies. Now, another thing that we did was to collect in the field urine concentrating ability, here measured as maximum urine osmolality in foxes, and plot them in the perspective of all the desert and non-desert mammals for which there's data on urine concentrating ability. And the values provided in this study for North African Ruppels and red fox from Northern Iberian Peninsula are like plotted here and highlighted with the fennec fox, and 120 more mammalian species that were reviewed in this meta-analysis that I did for Mammal Review. And what you can basically see is that both desert-dwelling species, Ruppels and fennec, here identified as Vulpes ruppelli for Ruppel and Vulpes zerda for fennec, are much higher than Vulpes vulpes, which is the red fox. So I guess to conclude, we basically found that ancient introgressive hybridization events or shared ancestral polymorphisms involving individual genes or structural rearrangements combined with independent selection on adaptive tolerance to extreme temperatures and increased capacity to preserve water seem to have enabled all these North African fox species to survive in increasingly arid desert regions at different evolutionary scales. And so this study is now successfully published in Nature Ecology and Evolution, and I'm showing here the cover of the issue that we were featured on, which was like huge. And so on a broader sense, on the origins of adaptation, if you ask me what this study is useful for, I would say that lends itself to the growing body of research that shows that interspecific gene flow and ancient polymorphisms involving structural rearrangements, they are actually very pervasive in nature, and they can be drivers of adaptive evolution across speciation events. If you think about it on the climate change perspective, desertification of arid regions worldwide are expecting to dramatically increase, and this may further intensify pre-existing selective pressures on desert dwelling systems. And so, based on these results, what we expect is that ancestral standing adaptive genetic variation that was inherited either via introgression or via ancestral polymorphisms seems to be crucial for ongoing or future adaptation to rapidly changing climates. And so ultimately, if I had to sum up the contributions that my work on the general theme of life in deserts and the genetic basis of desert adaptation, well, I have found throughout my PhD work and my early career scientist work is that we were able to identify shared patterns of adaptation in different biological systems with different ecological niches and at distinct stages of adaptive evolution in different deserts of the world. So there are shared patterns of adaptation in the different deserts of the world. We also integrated literature on physiology with bioclimatic variables and phylogenetic comparative methods, which is the second paper in Mammal Review. And here we found that aridity has been one of the main selective pressures driving phenotypic evolution in deserts. And then, most importantly, we set off to the Sahara Desert to study the evolutionary mechanisms driving repeated and independent adaptation in North African foxes. And with this multidisciplinary approach that it's far from being perfect due to, of course, the logistical challenges of sampling in North Africa in the deserts, we were still able to address outstanding questions concerning the origins and genetic basis of adaptation in arid regions and on the evolutionary history of these charismatic species. And so before ending my presentation, I would like to briefly thank both my advisors, Raquel Godinho and Rasmus Nielsen, and the past and present members of their groups, my institutions. At the time, I was being hosted and funded by the Foundation of Science and Technology in the University of Porto, and also by the Department of Integrative Biology at UC Berkeley via Rasmus, and in Porto via Raquel. But now I actually am a full integrative biologist department member at UC Berkeley, and you can find me as a really proud member of the Sudman Lab. So if you ever need, if you are wondering where I am, where to find me, and how to reach out to me, these are my current lab mates, and they've been a tremendous support, mostly because most of this work was finished when I was in the Sudman Lab. And of course, thanks again, Mateus, for inviting me and giving me the opportunity to talk and present a very brief overview of my work. It was a pleasure, and whoever is listening, I hope you may have found this useful. Thank you so much.
Wow, Joanna, I'm pretty sure everyone who will watch this will find this information very useful. You know, during your talk, I was just thinking how privileged we are, because, being a PhD student is not easy, right? We know that. We have several challenges in several fronts, but at the same time, there are moments in which you realize that all the struggle is worth it. When I was here sitting and watching you present and thinking, how else would I have the opportunity to, on a Monday morning, to learn about all these intricacies of the adaptation of life to the deserts? So yeah, thank you for sharing this wealth of information. Your talk was great. And I do have a few questions. I must warn you though, although I work with bioinformatics and genomics, I do not use your tools. So very likely my questions are going to be very basic. So, but I do have a few questions. When you were showing us the signs of introgression between the fennec fox and the North African red fox, you mentioned that you found that region of 25 mega base pairs long, right? And you mentioned, if I'm not wrong, you mentioned that this is an unusually large region.
Yes.
Is that the case? So usually when we see introgression, clear signs of introgression between species, how large are those introgressed segments, let's say?
Yeah, so just a quick correction. It was between the fennec and the Ruppel's fox.
Okay.
So when you do this kind of tests called D statistics or FD, you are normally doing them on a genome-wide level. So you are thinking about things that probably are less than a 5 megabase kind of way. And even so, these kind of like unusually large segments being highlighted as an outlier for these statistics on Windows or for either these statistics on Windows or FTs, F statistics on Windows, whatever you do, when you see these kind of outliers, you need to interpret them with cautious, because they can also be something else. What these are showing you is not so. On a global genome-wide perspective, those you can confidently say at a genome-wide level that there is integration when you do this without looking at specific windows, without specific outliers in mind. When you try to grab those outliers, you need to do some further investigations. You cannot just rely on them. You need to do some follow-up steps of analysis. So one of them, for example, is this one where you see the fennec and the Ruppels from the genome-wide tree. And one thing that you would expect if there was some sort of integration is for this tree topology to change. And suddenly this means that at that region, Ruppel's fox and fennec are more closely related than what you would expect from the genome-wide perspective. And indeed, if you look into this affected region, Ruppel's fox and fennec are more closely related. But that could also be because of something else. It could be because that region was maintained throughout the evolutionary history of this genus. And so it was something that was predates all the origins of the vulpes, and somehow it was something that is shared between fennec and Ruppels that may have been lost in all the other vulpes that are in the vulpes tree in between. And so there are follow-up, and perhaps the most decisive analysis is what you call the DXY. So if you knew for sure that this was integration between fennec and Ruppel's fox, what you would expect was for this DXY statistic for the affected region to be extremely low compared to the genome-wide. And this is not the case. So one of the reasons we think that this was not integrated, one of the reasons that when we see this, we cannot say, yeah, it's an integration case between fennec and Ruppels. When you see this, you can say, maybe this was not integration between Fennec and Ruppel's fox. Maybe this was integration between a sister species to the Fennec and Ruppel's fox, because these are already very divergent. Like there's not much difference in divergence between the affected region and the genome-wide region. So this means that either this was not integrated from the Fennec, but either a Fennec-related donor, or this was maintained across speciation events because it was advantage in a desert context, and it was lost throughout the Vulpes phylogeny in other species. But of course, to be fully sure and be able to discriminate this analysis, we would need to include a species-specific approach where we would have more Vulpes species in this work, and then we would be able to test, for example, okay, is this region, if we do this tree and we will include in this tree all other species, do the Ruppel's fox and Fennec still outgroup the red fox and everyone else, or does something change? And so there's some missing elements here that would allow us to reconstruct the full puzzle. So this is a really complex region. It would also help if we would have like long read sequencing and better genomes to kind of understand the extent of recombination suppression in the region, to fully understand if it's a structural variation case or what is going on. For sure, it is an amazing result. But one of the things that we tried to be cautious in this paper was to avoid overinterpretation. So we never say this is integration from Fennec to Ruppel's. We say that Ruppel's and Fennec share this very divergent block, and either this very divergent block is shared directly between Fennec and Ruppel's, or between something that is very closely related from the Fennec that we don't know what it is, and Ruppel's. So there's a kind of speculation element here in how we interpret these results. Yeah.
No, and I understand the need to be cautious about those conclusions, and I appreciate your explanation there. Another thing I was wondering is that, and again, this comes from the fact that I am totally alien to this field, is that would we care about the number of chromosomes those species have? Meaning that if these species have similar numbers of chromosomes, I'm assuming that it would facilitate introgression and assimilation of foreign DNA? Is that a thing?
Well, I don't know how the two relates, but for sure the thing is that integration is very pervasive in canids. And you see a lot of cases of not in the Vulpes phylogeny that is not very well understood. So I think we were the first to address this to the best of my knowledge. But in the Canis genus, so the genus that includes dogs, wolves, coyotes, the dhole and the jackals and the wolf. Oh, I think I said wolves. But if you look into the Canis phylogeny, which is the example that I give in my introduction slide, yeah, here you go. You see, like these are cases of species that are quite divergent from each other, and there have been identification of like integration across the entire regions and the entire lineage, sorry, and even from unknown species like a ghost lineage or as yet identified canids. And the truth is that all these species, so all the canids from foxes to dogs and wolves and the African hunting dog, they all have very different karyotypes and very different chromosome numbers. Like even within the Vulpes genus, they have very different chromosome numbers. Some of the foxes have, for example, B chromosomes, supernumerary chromosomes, which is something that I think the raccoon dog also does. So it's the entire canid family is very interesting in how diverse karyotypes they do, and that does not seem to be either. I don't know if that's a facilitator or of integration or not. For sure, it seems to be integration does not seem to be interspecific gene flow does not seem to be limited by karyotypic differences. That's what I could say. Yeah.
Wonderful. Yeah. In other words, I mean, the group as a whole is a great model system that it is understudied. And I hope that, with your research and your talks, more people get interested in the group. One last question. So when you showed us, you have several beautiful Manhattan plots, in which, some of those you used to show us. So I think you mentioned regions that are highly divergent, but at the same time, I think they exhibit lower diversity. Is that right?
So that is okay. Yeah. So I can go to that slide. So those are like when you're interested in finding signatures of selection, and I think
like this one,
that
yeah.
yeah. So there are many signatures or footprints of natural selection in the genome, and many people do this kind of selection scans. So one that is very common to do is FST based tests. So there's this idea that if you do selection and if a gene is under selection in a focal group, it will be very divergent compared to the genome-wide average in the focal group compared to a sister group and an out group. That is called the PBS statistic. So essentially, you're trying to look for regions that are highly differentiated in the population that was targeted by natural selection compared to the genome-wide average differentiation in the comparison to another to a sister group and to an out group, and that is a signature of natural selection or a selection test that you can use. But there's another one, which is another signature, is that if genes were selected or if mutations were selected, you would expect them to show reduced diversity at that region compared to the genome-wide. So it's a combination of two things. It's very differentiated compared to the genome-wide when you compare it to other species, but it's also reduced less diverse. So it's these two metrics that you can use. And we did this for North African red foxes compared to Ruppel's fox and the Arctic fox that we use as an outgroup. So, and the Manhattan plot that you're seeing here, so all these genes that you're seeing here, one in the upper one, they are highly differentiated in North African red fox compared to Ruppel's and the Arctic, compared to Eurasian red fox and Ruppel's as the outgroup. And they are and they have reduced diversity in North African red fox compared to Ruppel's and the Arctic fox. So yeah, that's the analysis.
Mm-hmm. And those points, so you're showing us different genes that you were able to find the signature for selection, but in the plot there, so the plot shows us a specific region that within that region you find those genes, or the plot is actually showing the gene SLC12A2 is in that.
The plot is done on sliding windows. So what
Okay,
you're
yeah.
seeing is like a windows of well, actually, I no longer know, but I think it's like 50 kb windows overlap by every 10 kb. Anyway, I think those details are in the paper. Yeah, I to be honest, I no longer remember what window size we chose, but yeah, and then those windows overlap with genes, so then you know the coordinates of those windows, you know the chromosome, the start, the end of the window, and then you can basically intersect with the notation of the genome, and you can understand where if there's a gene overlapping in that region or not. So basically what this shows is that these are extreme values of an empirical distribution, and our top hits, our top windows overlap with candidate genes that you identify by intersecting your windows with the annotation of the genome, basically. I hope I'm trying to explain this in a way that is not like super technical, but it's also, you know, like it's I hope I'm doing a good job here.
Oh, you are. I know the struggle. I know what you mean, and you don't want to be super technical, but also you don't want to say oversimplify and say wrong things. And I think
Yeah.
you deliver the message. No, I do understand. I actually forgot they were sliding windows. You're right.
Sliding window, North African red fox as the focal group, Eurasian red fox as the sister group, and Rupels as the outgroup in this analysis. Yes.
Wonderful. Well, I think that's enough of questions. You know, you've been talking already a lot, and I've been asking you a lot of questions. With that, I would like to thank you very much for your time and your attention today. And I always like to finish by asking, is there something, you know, I didn't ask you and you would like to talk about, like, or perhaps for another talk? Is there something you would like to discuss, or?
Oh, I guess for PhD fellows out there, I just wanted to let you know that PhD candidates are early career scientists. So I just wanted to let you know that, of course, this work was it involved very different stages. It included intensive field work, a lot of wet lab, and a lot of computational analysis, and building this entire project. I wasn't alone, so I'm speaking here as the lead author of this work, but of course I did this work with an amazing team of people. First of all, I couldn't have done this without the computational and experimental design approach that was pretty much, and the selection analysis and all the computational steps, and also like our low coverage approach. Everything was designed and discussed with my two advisors, and they were during my time at Berkeley was when I did most of my computational analysis, and if it wasn't for Rasmus, I would have not learn any of these that I'm trying to explain to you. And Raquel, of course, provided all the conditions for me to go to the fields and the physiology part, and granted me a team that included a wildlife veterinary, a fields technician, all the Sahrawi guides that helped me do work in the desert. So I collected most of this data. I went to the field, I extracted the samples, I did the analysis. It was a really intense and thorough beginning to end hands-on analysis. And I think that, yeah, it was sometimes was lonely, sometimes was frustrating, but I had an amazing team of from the advising, supervision to, of course, collaborators and contributions. So I just wanted to make clear, if I sounded self-centered or individualistic doing this, that of course I'm assuming that people know how science works, and that I'm standing on the shoulders of people that helped me so much and that taught me so much, and I'm so grateful for them. So yeah, that's the only thing I wanted to disclaim, I guess.
Sure. And I think it's a great way to really to wrap things up, because like you said, especially for early career researchers, meaning, you know, PhD students that are starting off, when we see successful PhD students, you know, successful PhD graduates, sorry, And I agree, just by watching your talk, I could envision the sheer amount of work that went behind it. But it's if you aren't told about the difficulties and about how many people were involved, you can have wrong guesses, right? And you can think like you can start comparing yourself to others. So it's good
Exactly.
that
I'm a mortal. I'm only human, and I'm not that brilliant or outstanding.
but your work is beautiful. So I agree with you that there was a huge team with you and you had good supervision, which is fundamental. But definitely, I mean, your hard work also paid off. So, well,
Yeah.
with that, Joanna, I would like again to thank you for your time. I feel really privileged to have the opportunity to learn all these things about desert life with you today. And I'll close our recording in a second, and I will as well. I'll let you know when this talk goes live. Okay, so just I will stop the recording now, and then we can chat a little bit more. And
All
for
right.
whoever's watching this, thanks for being here with us. Did you want to say something, Joanna?
Yeah, no, I'm fine. Thank you. Thank for whoever is out there
Watching.
listening.
Exactly.
Yeah. And
Thanks,
if I
everybody.
said anything wrong or bad, like I mean, the paper is just text me or email me or any question. Feel free to reach out to me, and I'm happy to explain better or provide any more detail on it. So,
Mhm.
yeah.
Good stuff. Thanks, everyone.